Yeah? I think I have too. I've got to go home and write an exam tonight. I know that breaks your hearts, but it kind of breaks my heart because I've got a very busy night. What's that? Yeah, I've had several offers already, actually. I'm letting you guys help to write the final. What do you want? I mean, you know. So, questions? Yes?
What protects the bond from water after
Oh, very good question. So, he's asking a question about translation and what protects the bond between the tRNA and the amino acid after the EFTU leaves. So I'll remind everybody that bond between the transfer RNA and the amino acid is very unstable in water. And that's one of the functions of EFTU, which is to protect that bond from being exposed to water. So I told you how EFTU carries the transfer RNA with the amino acid on it to the ribosome. And when it gets to the ribosome, the EFTU makes sure that the proper base pairing is occurring. If not, it takes it away and still continues to cover up the bond. If it stays there, and if it's there and it sees that the base pairing is proper, then EFTU leaves. And his question is, what happens when EFTU leaves? Wouldn't that expose it to water? The answer is it doesn't, and the reason is because the ribosome covers it up. So it's a pretty delicate balance that has to happen in this process. Otherwise, we would be losing that amino acid during that process. Very good question. Yes? Okay, can I go through the blue-white screening and talk about what caused it to turn blue? I can do it with a figure. How about that? It'll make more sense if I do it. Okay. So, and blue-white screening. Okay. So, we're doing blue-white screening for the following reason. All right? We want to be able to tell not only which cells get a plasmid, but which cells get a plasmid that have an insert that we've tried to put into it. That's the two things that we're doing. So blue-white screening helps us to see which of the plasmids have an insert. Okay? Is there a question?
Yeah. So in the middle one right there, it says that that's the one that turns the kind of little product.
Uh-huh.
This
one here?
Blue. Right. But the inner section is still intact, and I thought they would only turn blue when the lactase gene was cut.
It turns blue when the lactase gene is intact.
When it's intact.
Yes. Okay. So let me give some build up to that. But you've got it. Yeah. So yeah. All right. Now, what's happening is that when I cut, so let's say I want to put an insert into this plasmid. I want to put a gene into this plasmid. And so I would cut in the middle of this LAC-Z gene. That's the gene for beta-galactosidase. That's called LAC-Z. Okay? Well, LAC-Z gene, when the enzyme that is coded for by the LAC-Z gene is made, the enzyme will cleave this artificial substrate called XGAL and produce a blue color. So if you have a functional LAC-Z gene, then that functional LAC-Z gene will produce a blue color when treated with XGAL. Okay? That's the core of this. Now, I want to put my insert into here, so I cut the middle of the LAC-Z gene. I've got two possibilities. I can, in one possibility, get my insert in the middle of the LAC-Z gene here, or I can not get it in which case it'll simply, the LAC-Z gene will just come back and ligate on itself. Well, when it ligates on itself, it restores the LAC-Z gene. All right? However, when there's an insert, the insert interferes with the coding for the LAC-Z gene. It doesn't make a functional LAC-Z gene. So what that means is that this guy over here has an insert. When I treat it with XGAL, it's going to produce white colonies. There's not going to be any LAC-Z being produced. Those plasmids that have a functional LAC-Z gene, which is what happens when it religates without an insert, will produce blue colonies. And those cells that don't get a plasmid, of course, are going to die because I'm growing these on antibiotic, and my plasmid has an antibiotic resistance gene in it. So those are the three possibilities, and I can immediately look at the color and say, well, these guys I'm interested in because they've got inserts. These guys clearly don't have inserts because if they did, they wouldn't have a functional LAC-Z gene. Does that make sense? Does that make sense to you? Okay. It's a cool technique. I always like to say biochemists and biotechnologists are lazy. So if you find an easy way to do something, that's always preferable. So, question? Very good question. So her question concerns histidine tagging, which I think is further down. Yes, here. And her question is the histidine tag something that I'm adding to it? I'm adding to it in one sense. Okay? I'm not adding to it after it's been made, no. Because I wouldn't have a way to select only my protein to do it. But I am adding to it in the plasmid that I put it into. So here's an expression vector right here. And this expression vector has in it already a sequence of histidines at the very beginning of it. So when this promoter that's down here starts transcribing it, it transcribes not only the gene that I'm interested in, but also the histidines fused to it. So the protein that'll be made from that will have the regular green protein that's there, but it will also have those little histidines that I put on to the very tips of them. Okay? Those histidines then, when I load that onto a column, the histidines will stick to the column if the column has nickel in it. And the proteins that don't have histidine, which are all the rest of the cellular proteins, won't. They come shooting through, as you see here. Then, only after I add imidazole or histidine, and the same thing here, will I get my proteins that have the histidine tag to come off the column. So the beauty of this is in one step, I get the things that I'm after in contrast to the things that I don't want. Does that make sense? But yes, I do put it on, but I don't physically put it on. I make it part of the plasmid is where it's coming from. I'm sorry? So by the time it's already made as a protein, it's already here. It's already on the protein when the protein is synthesized. It sticks when I add it to a column that has nickel in it. So the histidines have very great affinity. And it takes a bunch of them. You can't just have one or two histidines. You've got to have a bunch of them clustered together. And you get that bunch of them clustered together, and they will stick to a nickel column. That's right. So I'm trying to separate the protein of interest away from all the other cellular proteins. Yeah. And then I can use a protease later to clip off the histidine, so I don't even have that histidine on there when I'm done. It's a pretty cool technique. Oh boy, so there's a good question. So her question is, are monoclonal antibodies only made in the laboratory, or do we make them in our bodies? That's actually a very difficult question to answer, okay? But I'm going to answer it, all right? So an antibody, a given antibody, okay? So let's say I have a cell that, or a B cell that has bound to an antigen and has recognized that antigen. That B cell will be stimulated to divide multiple times, and that all the products of that B cell will all recognize exactly the same piece of the antigen. Those will, in a sense, be monoclonal. Okay? So your body is making them monoclonally, but when we pull them out of the bloodstream, we get polyclonal because a different antibody that recognizes a different part of the antigen is mixed with it. Okay?
So the polyclonal antibody is
the mixture of all of the antibodies that recognize the antigen, but different parts of it. Make sense? Okay. And you had a question up here. Yeah, that's a pretty cool aspect about the capsid protein. Sorry, I say plasmid proteins. About the capsid proteins, they are literally like pieces of a jigsaw puzzle that come together, and they tend to have very regular shapes, so that you don't have to have an order in which they're assembling. So I gave the analogy, for example, of a jigsaw puzzle that assembles itself. It's not nearly so complicated for capsids. You could have, typically capsids of a virus, you may only have a handful, 4 or 5 of them. And they all have a regular shape, and they all have the right angles so that when they assemble, they start coming together in let's say a sphere is one example, that we might see them coming together as a sphere. And those pieces are able to literally connect with each other because it's like having a puzzle where all the pieces are the same. Okay? And the self-assembly, I'm exaggerating a little bit, but I think it's a good image for you to think about how a virus works. There are proteins that will help the capsids to come together. So since you've asked the question and you're not responsible for this, I'll tell you a brief, interesting component of that. Okay? So we talked about zymogens, right? And zymogens are proteins that are made in an inactive form. So capsids proteins are frequently made in an inactive form. And you want them inactive, not you, but the virus wants them inactive because they want to control when that assembly occurs. Right? So they want to make a whole bunch of these things and then assemble them. So they make them in an inactive form, and then they use a protease to clip off the part that stops them from assembling, and then they will start to assemble. All right? So when you hear HIV, and you hear about the treatments for HIV, one of the treatments for HIV are so-called protease inhibitors. And the protease inhibitors are there to stop the capsids from being able to form. Does that make sense? Yeah. Okay. Yep. Okay. So let me approach that by starting with PCR, if that's okay. Okay. So her question was she's confused about the terms I've used, vector, expression vector, and PCR. So let's talk about PCR first, and then I'll talk about expression vectors. All right. So the polymerase chain reaction is a laboratory tool. So too is an expression vector. Okay. But this is a laboratory tool, not to get a cell to do anything. It has nothing to do with a cell. This has to do with something I want to manipulate and I want to work with. So this would be done in a tube of some sort, and it's being done with the proteins that can help to replicate this DNA. It's a way of making more DNA. So I want to make a lot of a specific sequence of DNA. I would use the polymerase chain reaction to do it. That's what the polymerase chain reaction is there for. Okay? So the polymerase chain reaction requires several things. It requires a target DNA. It requires primers, and primers are necessary for the DNA polymerase to start replication. So it's got primers there and there. It needs a DNA polymerase, and it needs the 4 DNTPs. Okay? Now, go ahead. Yeah. Other ends, fine. So her question was, are there names for the primers? And for our purposes, the name of the primers will just, no. We'll say one versus the other. Okay? They're not the same. One's at one end, one's at the other end. And the primers are really defining the ends, the bounds of where replication occurs. Okay? All the replication is essentially going to occur between those 2 primers. That's why the primers are important, and that's why this process is really interesting, because it's amplifying not all the DNA in the cell or in the sample. It's replicating only the part that you tell it that you want to replicate. And that's what's happening as we move down this chart. The beauty of the polymerase chain reaction is every time we do a cycle of 3 reactions, we double the amount of DNA. That means we can make billions of copies of DNA in a very short period of time. Literally in 2 hours, you can make billions of copies of an original target DNA. Now, an expression vector is used for something completely different. All right? An expression vector is used by a scientist to get a cell to make something. It's used by a scientist to get a cell to make something. All right? What's it using? So if I go to my expression vector, okay, an expression vector has several things that are necessary for it to express. So the expression vector I'm using to get a cell to make protein for me. I'm getting a cell to make protein for me. That protein might be human growth hormone. That protein might be insulin. It might be any variety of things that I want to use, but I'm getting that cell to make it for me. And the expression vector is used to make that happen. All right. So an expression vector, to have an expression vector, I must have several things. First of all, an expression vector is a plasmid. It's a plasmid. Second, that plasmid has an antibiotic resistance gene in it. Now that antibiotic resistance gene is important. Why? Why do I care that it has an antibiotic resistance gene? It's a marker that I can tell which cells get the plasmid, right? Okay. Third, the plasmid has a promoter. And the promoter is going to be necessary for me to put my gene in front of so that it gets transcribed in the target cell. Now, those three things are really all I need for an expression vector. I can put histidine tags in there. I can put the LAC-Z gene in there. I can put a variety of other things in there that might help me. But the three things I just told you are the minimum for a plasmid. Oh, well, back up. A plasmid, by definition, has a replication origin. Okay? So a plasmid has a replication origin. So if you want to count that as a fourth, that's the fourth. Yeah. So an expression vector is a plasmid, number one, has a promoter, number two, has a selectable marker, number three, and has a replication origin, number four. Yes? Let me make sure I understand. So you're saying if I were to make a protein like insulin in a bacterium, will it have any funky characteristics that might cause an immune response if I gave it to a person? Is that what you're saying? Yeah. Okay. So it's a very good question, actually. So the answer is we try to design these things to minimize that effect. That is, yes, if I were to take, for example, the insulin gene, and I were to give it to E. coli and say, make this insulin gene, and then I took it out of the E. coli and I tried to give it to people, I would have problems. And the reason I would have problems is because you may recall that there's post-translational modifications that happen to insulin. Okay, that's not it. Where's it at? Okay, that'll do it. Okay. So there's post-translational modifications that happen to insulin. A bacterium doesn't have the ability to do that. So ideally, if I want to do this, I would put it into a cell that had the ability to make those modifications, or I would make those modifications separately. Now, your question was, is it possible that I would have some things that might come along for the ride that would cause an immune response or something if I did that? The answer is potentially yes, but in reality, many of the recombinant proteins that are used medically have reduced the problems associated with the immune response. And that's because in the past, they had to take them from other animals to get them to do it. So for example, if you wanted to have, well, an example I think I gave in class was insulin itself, that they had to isolate it from cows. And it's not an identical insulin to human insulin. So when they put cow insulin into people, it works a little bit at first, but over time they develop an immune response to it and thereby lose the ability to have that function. So if you give human, and it's properly modified human insulin to people, the likelihood of that response being an immune response goes way down, way, way down. And the only way you would get a response would be either if you hadn't processed it properly or if you had some contaminating things that came with it. Make sense? Good question. Yes? Say that again, I'm sorry. Okay. Okay. So she said in her notes it says that NAD to NADH is a catabolic process. And I would say, no, that's not quite correct. So let's clarify that for you. And I talked about that when I talked about energy. Okay. So when I look at oxidation reactions that happen, okay, here's an example of an oxidation reduction reaction. Okay. Going rightwards, I've got oxidation going on. Going leftwards, I've got reduction going on. All right. Oxidation reactions will tend to be catabolic, and reduction reactions will tend to be anabolic. Now that's where that's where your question is sort of related to that. So if I were going through this process in my body, when would I be going through this process in my body? After drinking a beer, right? All right. So if I go rightwards, all right, I am going to oxidize this guy back to acetaldehyde. What am I going to produce? Well, I'm producing 2 electrons and 2 protons. And in my body, I said we don't have free electrons and protons. So what happens to them? They get taken up by an electron carrier, in this case NAD, and making NADH. So I could take ethanol plus NAD goes over to this guy plus NADH. So the catabolic process, NADH would be a byproduct of it, but NAD to NADH by itself in isolation would not be a catabolic process. Does that make sense? Okay. So a catabolic process will produce electrons generally, and it will, as a consequence, need NAD to accept those electrons in the process becoming NADH. Yes? So NAD is the oxidized form, NADH is the reduced form. That's right. For every oxidation, there's an equal and opposite reduction. Okay, so her question is the difference between a Hayworth and a Fisher. And if we look at, yeah. That's, well, let me just go back to the figure that you're talking about. So if that's not what I want. Okay, so this is what some people would refer to as a Fisher structure. This is what I refer to as a Fisher structure. Okay, so I don't like that designation. This to me is a sort of a modified Hayworth. Okay, so a Fisher projection is linear, a Hayworth is a ring. Yeah, this is just a dumb designation in my opinion. Yeah. For what that's worth. Okay. I can assure everybody I will not ask you to draw one of these. Okay. So her question has to do with sugar alcohols, And it looks like on the example that I gave that they came from ketones. And if I can remember where that, here it is right here, alditols, okay. So the examples, sorbose is going to sorbitol. But you could also get to sorbitol if you started with an aldehyde up here. So it doesn't have to start with a ketone. I could reduce an aldehyde and also produce a sugar alcohol. So in other words, if this were an aldehyde up here and instead of a ketone, and I reduced the aldehyde, I would get the same thing.
But is it the same? Is it a different sugar? Is it still
Nope,
sorbose?
it'd be the same thing. Well, I mean, sorbose is a ketone, so I mean, I can't say that, but this sorbitol could be produced from either an aldehyde version of this or a ketone version of this, is what I'm telling you. Okay? So the examples that I have just related, just showed ketones, but I could reduce glucose, for example, and make glucitol.
Right.
Yeah. I'm sorry? A sugar alcohol is a reduced sugar. That's correct. Yeah, and a reducing sugar is a sugar that can be oxidized. It sounds like they're all contradictions in terms, but it's how you say them. Yeah, but you said it exactly right. Yep. So a reducing sugar is a sugar that can be easily oxidized, whereas a reduced sugar is something that has been reduced. Yeah. Other questions? Yes? Yes? Okay. So our question is, I talked about proto-oncogenes and oncogenes, and I talked about mutations in proto-oncogenes, and your question, I think, was where does that sort of occur in the scheme of things? Is that right? Okay. So I haven't really given it as a progression of events, but I will briefly tell you that mutating a proto-oncogene is not something you want to do, okay? Because mutating a proto-oncogene, it doesn't always happen, so it depends on where the mutation is. But mutating a proto-oncogene can convert it into an oncogene, all right? So mutation, when is mutation going to happen? Well, it's going to happen during the process of replication, or it's going to happen as a result of damage to DNA. So a real prime time when that damage might occur would be, a tanning booth, cigarette smoke, contaminated water, contaminated air if you're breathing a lot of exhaust fumes, contaminated food if you're eating a lot of pesticides. All of those things could lead to damage to your DNA that would result in mutation. In fact, all those will result in damage to your DNA and will result in some mutation. You've got mutations going on in your body right now constantly, all right? People have these real big notions about, I'm going to detoxify my body, you know? And it's like, get real. It's not going to happen, okay? I'm not saying you should be eating things and drinking things and smoking things that cause problems for you like that. But at the same time, there's no way that that these notions of toxins that people have that they're going to get rid of, because they're not. Okay? So to come back to your question, when does this happen? It's happening as we're sitting here. It's happening as you're completely unaware of it happening. So the more exposure you have to what are called mutagens, and all these things I've just described are mutagens, they're things that favor mutation. The more exposure you have, the more likely you're going to mutate. The more mutations you have, the more likely you're going to mutate an oncogene. So it's all a numbers game at some point. It's probably not the answer you were looking for, but yeah, that's what it is. Other questions? The functional difference between the light chain and the heavy chain in the antibody, I haven't given one. So since you've asked the question, since you've asked the question, again, I will briefly tell you, but it's in the heavy chains where the differences are between the different classes of antibodies. Okay? But that's more than we need to know for this class. Mm-hmm. Yes? Yes. Good. You know, I did too. I always get excited about that. You, yeah. Yep. And then, well, okay. So let me go to the figure, which I still haven't corrected. I need to do that. All right. So, in going from 3-phosphoglycerate to 2-phosphoglycerate, all right, what happens is we start with this guy. All right. This enzyme is a mutase, and mutases work not by moving things from one to another, but by adding a second copy of it and then removing the first copy. So there's an intermediate that's made where this 3-phosphoglycerate gains a phosphate on position 2. That makes 2,3-bisphosphoglycerate, and then the 3-phosphate is removed. So you're left with only the 2. The enzyme name, yeah, sure. Because I said you should know the names of those enzymes. Yeah. So our question is, in making a glycoside, I said that we altered the hydroxyl on the anomeric carbon. What replaces it? It really varies a lot. So you could, for example, replace that hydrogen with a methyl group. You could replace it with another sugar that might bind there. You could replace it with a phosphate. All of those would give you glycosides. So if you alter that hydroxyl on the anomeric carbon in any way, you create a glycoside. Any of those would do that. Yes? Yeah. There's no what? Yeah, right. Okay, very good question. So her question is, I said in the replication of HIV with reverse transcriptase that there's no proofreading that occurs, which is why the replication process is so so changing. That is, it's not very accurate in its replication. And our question is it because that the 3' to 5' exonuclease is inactivated, or is it because it's not present at all? The answer is it's not present at all. Well, before you finish, I will say one thing, and that is I have a very busy day tomorrow, so if you're looking for me tomorrow, I may be hard to reach. I'm going to be over, tomorrow there's a big event over in the Quad called Celebrating Undergraduate Excellence, where students are presenting posters of their research work. I'll be at that because I'm running the event for most of the day. So 2 things. One is if you catch me there, I'm happy to answer a question if I can answer it for you. And 2 is I will be in my office all day Wednesday morning, with the exception of after about 10 o'clock. I've got a meeting until 10. After 10, I should be in my office all day. Okay? All right. Good luck. Study hard. And let's see you guys do like you did on the last exam, a nice average on the exam.